and I want to talk about metabolic pathways. As chemicals are being changed, as anabolism is happening, catabolism is happening, things are changing, you go from one molecule to another. And we call these biochemical pathways or metabolic pathways. You start with one thing, you end up with another. Now, when we get into this, they're going to have proper names and they're going to get confusing. So I want to take a minute to take it back to the basics. And so no matter how crazy it gets later on, just come back to this. Is this a precursor? Is it an end product? So come back to this. So let's look at what I have drawn. All right, I always think it helps you to draw it too, so go ahead and humor me, get out your pen and paper and kind of draw it too. I know you got a screenshot. It just makes our brain learn it better, so draw these things out with me. So I am going to draw a representative metabolic pathway. I'm starting with one molecule, that's A, and it's going to go through several steps, and it changes a little bit at each step, and we're going to end up with molecule D. So each one of these molecules is going to have a proper name, but we can also give it a name in relationship to the pathway. Just like my name's Tiffany Martin, but I'm also a professor. I'm also a mother. I've got a name based in a relationship to what's going on and what's surrounding me. So this starting molecule is going to be called a precursor. So any molecule you're starting at the beginning of the pathway is going to be called your precursor. What you are trying to end up with is going to be your end product. Okay, so you start with a precursor, whatever its proper name is. In this pathway, it's the starting molecule, and so it's called the precursor, and whatever you're ending up with is called the end product. Anything else in between, and it could be one thing, could be a whole bunch of things, we're just going to call those intermediates. We weren't trying to make them. We just made them in the process of getting to our end product, which was what we wanted. So B and C are intermediates in this pathway. So we've given it a name. All right, so basically what I want to do is I want to take molecule A and turn it into molecule D. So there's some steps that happen along the way. So to get from A to B, I'm just going to change the shape of it. I'm not going to take anything out. I'm just going to kind of rearrange the carbons or hydrogens, oxygens, whatever it's made up of. I'm just going to rearrange it to get something different. Now, this could happen spontaneously. We waited enough time. It could happen. But we want it to happen fast. This is metabolism. We're trying to build stuff. We want to make it happen quickly. So in order to do that, I'm going to use a helper. I am going to use an enzyme that is going to take molecule A, hold on to it, and rearrange things until it looks like molecule B. And every time enzyme 1 binds to molecule A, it is going to do its job. It's going to change it until it looks like B. That's its job. That's the only job it does. It does that over and over again. Enzymes are very specific. They can only take A and turn it into B. Now, I need to keep this pathway going because I'm trying to get to D. To get from B to C, can I take enzyme 1 and try it, and have it go change B into C? As you would imagine, answer is no. I need a different enzyme. So we're going to call that one enzyme 2. It's a different enzyme, has a different job. Anytime it sees this molecule B, it's going to bind to it and change it into C. So every step along this pathway is going to need a unique specific enzyme that's its only job. Now this time when we're going to B and C, we actually see it just kind of looks like we cut off a piece. I know I could draw it better, but I can't draw. Are you visualizing my cows from previous lectures? Anyway, it looks like this little end piece was kind of come off, and you can kind of see it down here. I'm going to call this F. It's probably made up of some carbons and hydrogens or, whatever, I'm not sure. But either way, enzyme 2 took out some things to produce F. This is what happened in this step of the reaction. So B to C required removing a few things. So this one was just rearranging, this one actually removed it. So when we remove things, we've made a product, we made something, but it's not the end product. It's not what we were trying to make. We weren't trying to get F, it just appeared in my process. We've got to give that a name, and we're going to call all of those things byproducts. These are molecules that are made as we're going down the pathway. So F is an example of a byproduct, and I can actually show you some. You ready? Here comes my byproducts. Can you see? These are my byproducts of cellular respiration. This is my body taking glucose, breaking it down. It uses oxygen during that process. What it makes is going to be carbon dioxide. I wasn't trying to make carbon dioxide. It was a byproduct of this reaction. And so byproducts, typically your body has to get rid of it. So we breathe that carbon dioxide out, and it's considered a byproduct. What else? Water is also a byproduct of cellular respiration. So when I undergo that process, there's a little bit of water that comes out. Aren't you glad the classes aren't live for people in the front row? A little bit of water and also some heat. Remember we said when things get broken down, catabolism, glucose is getting broken down to make ATP. Not all of that energy can be harnessed, and so some of it gets lost by heat. And so when I do this, are you doing this at home? Are you trying it? Okay, when I do this, it gets a little warm. There's some heat generated from that. All right, so that's some byproducts of cellular respiration. But in general, anything that you make in a biochemical pathway that you weren't trying to make is considered a byproduct and typically has to be gotten rid of from that. All right, are you with me so far? This really isn't bad, right? So why is everybody dreading metabolism? This is a representative of a biochemical pathway. All right, so we're going to add in a couple of things. So pretend your bacteria cell is doing well, and it's taking A and turning it into D, and D is what it needs, and everything's going well. It's getting rid of its byproducts. But suddenly, it finds itself in an environment where A is no longer present. A has disappeared, no longer has A, and it still needs to make D, but now we don't have any A to start with. Okay, think a minute. What can you do? Sometimes I hear students tell me that, well, we'll just start with B. Problem is that you need A to get to B. So that's a problem. So you still need D. You still need to make D. Sometimes we talk about in our study groups that D, maybe there's a different pathway. Maybe you can get D another way. And that's possible. There are some alternative pathways. But, you know, I'm kind of lazy, just like you. I don't want to do anything that I don't have to do. I want to get things going. I want to use the least amount of energy to try to get to D. And so sometimes what happens is A may not be available, but there may be a similar looking molecule that we can convert and take it and make B out of it. So A is not available, but I have G down here, and I can take G and turn it into B. And then what happens? B to C, C to D, and then I've got my end product, and my bacteria cell lives. It's very important to keep metabolic pathways going because many of these end products are crucial to survival. And if without them, you will die, and we don't want to die. So A is not available. Maybe A is glucose. Glucose is no longer available, but if a bacteria cell can take fructose, an alternative precursor, let's write that in there, it can take a different molecule and convert it into B, then crisis averted. We get to live. So G is an example of an alternative precursor that leads into our metabolic pathway and keeps us alive, keeps us producing end products. So you'll see that as we go through and talk about metabolism. Okay, hey, how does G get to B? So G to B, can I take enzyme 1 and just make it convert G into B? Enzymes are specific, right? So I'm going to need an entirely different enzyme to convert G into B. So I need a new enzyme to do that. So there's another enzyme, enzyme 4. Enzyme 4's job is to bind to G and turn it into B. Now sometimes these pathways have multiple steps. Sometimes there's a couple different pathways, molecules involved, but we're going to need some new enzymes to make that happen. Okay, so keep that in mind. Okay, let me give you another issue. So say you've been working and you need this end product and it's been working great, but suddenly a mutation happens or something changes, and now D, which was doing whatever job you needed it to do, no longer works. D won't get the job done. You're making plenty of it, that's great, but this D is no longer capable of doing what it used to do. Again, something's changed in the environment. There's a drug, there's a chemical. D is no longer being able to be used correctly. What's going to happen? You could die, right? This bacteria cell could die, and we don't want to die. So we need an alternative. If we had something different that could get the job done, then we're saved. I'm going to change our metaphor briefly into something that makes a little bit more sense in my mind. But you can kind of think of this process kind of as an assembly line, right? Say we're a company that's making bicycles, and we are going from A to B to B to C, and we're taking the bicycle frame, and we're adding the handlebars, and we're adding the wheels, and then we have a fully functional bicycle. And that's our company, and we're making money, and all that's great. And then suddenly we've got different people, different enzymes doing their portion of the assembly line. But suddenly nobody wants bicycles anymore. Maybe one of those talent shows came out and unicycles, TikTok, it's TikTok, right? TikTok, there was a unicycle video, and now the kids don't want bicycles, they want to buy unicycles. And our profits are being affected. Nobody's buying our bicycles, and we're going to die. We're not going to be able to pay our people. We're going to have to lay off. We understand that going on right now. So this is a problem for businesses. How do we solve this problem? Well, let's start making unicycles. If people want unicycles, I know how to make bicycles. Can I adapt quickly and make some unicycles? That's what people want. That's what people need. That will keep me in business. So let's make a alternative. I'm going to get rid of a little bit of this. Let's make an alternative end product. Something that is now going to be useful in our cell. And I am completely tired of drawing shapes, so we're going to call this one H. This again, D wasn't getting the job done. We want to try to make H's instead. Instead of bicycles, we want to try to make unicycles. Now, I could start from scratch making unicycles and develop this whole new process, but that's going to take too much time and money and resources and people and training, and I don't want to do it. If I know somebody, in my assembly line, people know how to put on wheels. Let's just retool some things and just put on one wheel instead of two. So make some modifications, and instead of ending up with D, we're going to do most of the work and then end up changing just slightly and end up with our alternative end product here. Now to do that, what are we going to need? We are going to need a new enzyme to take care of that. We may need a couple of new enzymes to take care of that. But this is a way we can stay alive when D isn't working anymore. And you will see this happen in metabolism as well if it produces an alternative end product. Again, thinking about drugs, this is how bacteria, we find a drug and that drug destroys D, and that's not working anymore. But if that bacteria can make an alternative that'll do the job that D was doing, then that drug can destroy D all it wants to, but now this bacteria cell gets to live. We get antibiotic resistance from that. So we're going to see examples of this as well as we go through. All right, but again, breaking it back down, just labeling it and its relationship from that. All right, the last thing I'm going to assign you with at the end is let's think about, I think I'm going to send you to your study groups to talk this out. Let's think about if you were a drug company, and this is a bacteria, and this is a biochemical pathway that only bacteria do, doesn't affect the host. Remember those magic bullets, chemotherapy? We had a magic bullet that would kill the pathogen without harming the host. So I know this is needed in my bacteria cell. I know that it doesn't hurt, that the host doesn't need this. How can I design a drug that will stop this biochemical pathway? Okay, what would you do? Where would you attack it? Would you attack the molecule, the enzymes, which ones? How would that look like? And remember, drugs are big business. It takes a lot of time and energy and money. It can take up to a decade to get a drug from idea to market. Clinical testing, trials, in vitro testing, tons and tons of effort goes into that, tons and tons of money goes into this. We need a fully functional drug. This is a big business, so you need to pick a good target to interrupt in this process. Okay, so hopefully you had some time to check with it. I might have done you a quiz. Maybe you checked with your lab groups and stopped the video and kind of thought about it for a minute. So what I hear a lot of when I assign this is that people say, well, first I have people that say, well, let's just get rid of B, or if we get rid of A, let's get rid of A. If we take that away, then we'll never get D. Problem with that is it's very hard to take away a whole nutrient that's found in the environment, and it's very difficult. How can we remove all glucose from the environment or something? It's very difficult. And plus we have this alternative precursor going on. So it's very hard to remove a precursor from this pathway. So that typically doesn't work. Let's look at enzymes. If I was able to bind an enzyme, if I was able to destroy an enzyme, if I was able to have a drug that would bind to this enzyme, number one, and keep it from working, that is actually a very good way, and a lot of the drugs work that way. They bind to these enzymes, and the enzymes are not able to do their job. If enzyme 1 is bound to this drug, then it can't take from A to B. So the next question is, which enzyme do you target? Enzymes are very specific, and so the drugs have to be very specific. I just can't wipe out all enzymes. That could interfere with a lot of other things. I need to target one of these enzymes. So if I did target number 1, that would work until it found an alternative precursor. And then the bacteria would live again, and that would not be a drug. This bacteria is now resistant to the drug that targets enzyme number 1. If I target enzyme number 3, something very close to the end, that also can be a problem because it's so close, one mutation, and we get something different, and we can use an alternative end product. So sometimes that's good. Typically, what drug companies try to do is they will target one of these enzymes in the middle. I kind of drew my X through number 2 there, and stop that enzyme. It's invested so far and it's still a long way away, and that's usually a good kind of target. All right, so these are real world problems. This is what people are doing on a daily basis. You've got to understand these pathways in order to happen. But when we start getting into respiration, come back to this if you need to kind of say, okay, all of this is kind of confusing me. What was that representative? Oh, it's just an intermediate. No big deal. Oh, it's just an end product. I see what's happening. Oh, it's making byproducts. So come back to that, and sometimes that will help you. Okay, I hope this was useful, and I'll see you at the next video.